Hello and welcome to Dr. Redford here, and today I'm going to talk to you a little bit about how inhibitors actually work. Okay, so first of all you have two types. You have non-specific and you have specific. Right, now the difference between these two is non-specific inhibitors are inhibitors which don't actually act on, that aren't specifically designed to act on the substrate, to act on the enzyme, in order to change its shape so that the substrate cannot bind to it. So these are things like temperature, and pH. Now pH you might not have heard of. It's on a scale from 0 to 14, 14 being something called alkali, and 0 being acid. Don't know too much about that for biology, it's mostly chemistry that is, but the point is enzymes usually work best around the middle of the scale, which is a neutral pH. So if it's too high or too low, the enzyme can become denatured. Now this word denatured you may not have heard of, but denatured just means basically unravelled. So basically the enzyme has its own 3D shape known as tertiary protein structure. I'm not going to go into that with too much detail with you to bore you in this video, but in another video I might make specifically on tertiary protein structure, so you can focus on that. But anyway, so that's the point. It can unravel the tertiary structure and become denatured if these conditions are not kept correct. The same goes for temperature. Obviously 37 degrees is the temperature of the body, so therefore this is the temperature that most enzymes function at their optimum, at their sort of highest rate. That means optimum just means highest rate, so right, optimum up here. If the temperature is too high, the enzyme again can become denatured. If it's too low, it means the activation energy isn't reached. So right, activation energy. Now activation energy is the energy required like from a collision between something called a substrate and the enzyme in order to initiate the reaction. So supposing you've got your enzyme here, and this bit here is its active site, which basically the site of the enzyme where the substrate bonds. Now the substrate can be this sort of shape, as long as it's complementary, which means as long as it fits into this active site, it will actually, it should initiate a reaction, but it obviously won't. Like supposing it doesn't have the correct activation energy or hasn't reached it, it means the collision won't collide with enough force in order for this substrate to bind to this enzyme active site in order to form something called an enzyme substrate complex. Substrate is anything that's broken down by an enzyme. So this can be proteins, it can be fats, it can be starch, it can be anything really. But the point is, unless this collision between this enzyme, between this enzyme and the substrate in its active site, unless there's enough energy there, it won't actually initiate this breakdown of the substrate. Okay, so that'll write down enzyme substrate complex. Now you have another two different types of inhibitors, and these are called competitive and non-competitive inhibitors. So I just wanted to mention those briefly. Now basically a competitive inhibitor is something which competes with this substrate in order to block this active site. So supposing this active site becomes blocked by some sort of competitive inhibitor, what this means is it means that this substrate or this molecule of some sort, so it could be protein, it could be fat, it could be anything really, but the point is that this cannot be broken down into smaller parts in order to be used by the cells in order to form energy that's required for respiration and other bodily processes. So the point is, our bodies cannot function very well if these inhibitors are blocking this active site of our enzymes. So basically, that's what a competitive one is. It competes with this for the active site, whereas down here you have something called the allosteric site. Now the allosteric site is a site, like in the back of it, so I'll know this allosteric site. Now the allosteric site is a site basically where non-competitive inhibitors bind. So non-competitive inhibitors. Okay, so the main point, excuse this noise for a minute. Right, the main point of non-competitive inhibitors is that they don't actually compete with this substrate for the active site, which basically means that they're not competitive in a sense. But when they do bind to this allosteric site in the back of the enzyme usually, this will result in the enzyme's tertiary structure tertiary protein structure becoming denatured or unravelled, which prevents this substrate fitting in here anyway. So it actually alters the whole structure of the active site, so it means it cannot bind. So I hope this video has been helpful, and that's a bit about the different types of inhibitors. So see you in the next video.